Hi, my name is Jackie Regensberger, and today I'm going to be presenting how the gut microbiome or viral infections affect brain development and function, and specifically between the gut microbiome and anxiety. So the itinerary for today is I'm going to be doing a brief introduction of this topic, some background information to make everything a little bit more clear, an introduction of the paper and what they will be researching, the methods that they used and their results, and then a brief discussion and conclusion of everything and different interpretations. So for our introduction, we're going to start with definitions. So first, anxiety can be defined as a complex emotional state characterized by feelings of unease, worry, and fear that are associated with specific triggers which genuinely vary person to person. The gut-brain axis is the bidirectional communication between the gut and the brain and has gained attention in various psychological processes, which includes anxiety. The microbiota composition is the gut microbiome. It's comprised of trillions of microorganisms and has been implicated in influencing brain function and behavior in relation to anxiety. The brain regions and cells involved is the amygdala, which also can be abbreviated as CEA, and processes emotions, including fear, and links them to memories and behaviors. The soluble epoxide hydrolase amygdala, also called SEHCEA neurons, are part of the amygdala, and they transmit signals related to anxiety and other emotional responses. They're both key components in the regulation of anxiety-related behaviors. So the relevance of this topic is that anxiety has a global impact. Anxiety disorders have a prevalence in our society and are the leading cause of disability. So it's very important to understand the neuromechanisms involved. A known aspect that we know is the CEA's role in anxiety regulation. We know that it does have a role, but what is unknown is how this mechanism actually works and the how it regulates this. We know that there is an enrichment of soluble epoxide hydrolase, SEH, in the CEA neurons, and it has a role in regulating anxiety-related behaviors in adult male mice. A little bit of background is there were key studies done. There was research by Dr. Kalin, and they highlighted the role of the central nucleus of the amygdala in anxiety regulation, which laid the foundation for further investigations into the neural mechanisms involved. Dr. Tai provided insight into the bidirectional communication between the gut and the brain, which emphasized the relevance of the gut-brain axis in influencing anxiety-related behaviors. Dr. Byrne revealed correlations between specific gut microbiota profiles and anxiety-like behavior, which shedded light on the potential influences of gut microbiome on anxiety regulation. All of these researchers found evidence from human, primate, and rodent studies and how it has implicated the central nucleus of the CEA and how it plays a critical role for the pathogenesis of anxiety disorders. So some more information to know is neurotransmitter production includes the gut microbiota, and they play a role in the production of neurotransmitters such as serotonin and GABA, and they're linked to anxiety regulation. And immune system modulation, which is the gut microbiome, and it influences the immune system and immune dysregulation and how it's related to anxiety disorders. So now introducing the paper, the goals was to find the role of soluble epoxide hydrolase in regulating anxiety-related behaviors, specifically in the CEA in adult male mice. It aims to address the elusive mechanism of anxiety disorders, which have been implicated in the pathogenesis of anxiety disorders in human studies and primate and rodent studies included. It aims to explore a previously unknown cellular and circuitry mechanism that guides SEH-positive neurons' response to anxiety, with the goal of revealing the role of SEH and its impact on anxiety-related behaviors. In order to advance the field, the study aims to do this by providing insights into the molecular and circuit mechanisms underlying the modulation of anxiety by SEH-CEA neurons and their projections to the bed nucleus of the stria terminalis, also known as BNST. So the hypothesis is that the SEH plays a crucial role in maintaining the excitability of SEH-CEA neurons and bidirectionally regulates anxiety-related behaviors, and it is predicted that the deletion of SEH in CEA neurons will induce anxiety-like behaviors, and that SEH-CEA neurons will have a direct projection to the BNST, which influences anxiety regulation. Now getting into the methods and materials. So animal models were used. The researchers conducted experiments using adult male mice to analyze the role of SEH in regulating anxiety behaviors. They used a brain tissue analysis where the scientists examined the composition of the gut microbiota in relation to anxiety-like behaviors in the mice. They used whole brain precise imaging in order to see the whole brain and visualize intricate connections across different brain regions. They performed behavioral tests where they used the EPM apparatus to assess anxiety based on the times traveled within the open arms, which is the white section in the picture on the right. They also performed neural circuitry analysis to study neural circuits and projections to understand the role of these neurons in anxiety modulation. It was found that SEH is enriched in the neurons in the central nucleus of the amygdala. It can be shown in the figure in the top left that all that green is representative of the SEH, and you can see that there is a lot of that in the first image. It was found that the deletion of SEH in CEA neurons triggered anxiety-like behaviors, indicating a pivotal role in SEH in modulating anxiety. The figure on the top right shows the results from the EPM maze, and it shows that the mice who spent more time in the open arm were very anxious. It was also revealed that SEH is required for maintaining the excitability of SEH-positive neurons in the CEA, and the bidirectional regulation of anxiety-related behaviors by SEH-positive neurons was demonstrated through chemogenetic manipulations, which highlights the complex interplay of SEH in modulating anxiety. This is because it regulates the level of acids, which are signaling molecules which have roles in modulating neuronal excitability. It was also found that the SEH inhibitor TPPU was shown to attenuate the intrinsic excitability of SEH-positive neurons and the CEA, which provides insight into potential therapeutic strategies for anxiety disorders. This can be shown by the figure in the bottom right, which shows that there's a decrease in firing frequencies in the SEH-positive neurons in response to gradually increased injections of the TPPU group compared with the vehicle group. It is also shown that the firing threshold of the first action potential is elevated by TPPU, which indicates a reduction in neuronal excitability. The study revealed that soluble epoxide hydrolase is enriched in neurons in the CEA and associated with anxiety-related behaviors, which indicates a pivotal role of SEH in modulating anxiety, and supports the hypothesis that SEH plays a role in maintaining SEH-CEA neuron excitability. I do believe that this paper is credible because all of the evidence is well supported by these experimental findings, and it provides valuable insight into how anxiety really is regulated in the body. Some future research could include the potential to create therapies to target the gut-brain axis, and it can lead to the development of a microbiome-based treatment for anxiety disorders. Thank you for listening, and I hope that this article really helped you learn something new.